The scientific name for a zucchini is Cucurbita pepo. They are a part of the plant kingdom, followed by the phylum Angiosperms. This is then followed by the class Dicotyledons. Then comes the order Cucurbitaceae. That is the family Cucurbits, then the genus squash, and the last category is the species zucchini. Zucchini perform basic systems of gas exchange the same way a lot of other leafy green plants do, through transpiration. There are stomata on the surface of the leaves of the zucchini plant. Their openings and closings are regulated by guard cells. If enough water is in the plant, the guard cells plump up, opening the stomata, allowing for evaporation of the water to occur. The opening of the stomata also allows for photosynthesis to occur. Light causes the stomata to open up a lot more because more light means more photosynthesis. If more photosynthesis is occurring, then more carbon dioxide must be coming in and more water vapor must be leaving as a waste product. When there is not enough water in the plant or sunlight is not available, the guard cells will deflate, causing the stomas to close. Zucchini plants are tracheophytes, meaning that they have vascular tissue and vessels to help transport materials throughout the plant. They have vascular tissue known as xylem and phloem tubes. The xylem tubes carry water from the roots all the way up to the leaves because of capillary action and transpirational pull cohesion theory. This theory says that when one molecule of water moves up the xylem tube, another water molecule gets drawn in from the roots. Phloem tubes are used to transport sugars from the leaves of the plants where photosynthesis occurs to all the rest of the plant. Because zucchini plants are planted in the ground, their roots extend into the soil. On the roots are tiny hairs known as root hairs. These root hairs greatly increase the absorption of nutrients from the soil and into the plant. The best nutrients for zucchini plants are potassium and phosphorus. The majority of the excretion in zucchini plants is done through the stomata as mentioned before. The waste products of photosynthesis are oxygen and water vapor, and both of these exit the plant through the stomatas. The purpose of gene editing on the zucchini plant is to protect their genes against certain viruses and to make them resistant against these viruses. This plant was genetically modified to be resistant against zucchini yellow mosaic virus and watermelon mottle virus 2. Both were developed by the Asgrow Seed Corporation. The transgenic method used to genetically modify squash is agrobacterium-mediated transformation. The tumor-inducing or TI plasmid is a type of bacteria that moves its transfer DNA, also called tDNA, to the nucleus of a plant. These genes can be transcribed, translated, and regulated. The genes in the tDNA were replaced with genes producing coat proteins of the viruses affecting the squash. The gene expression of the coat protein tDNA was regulated by a promoter 35S from cauliflower mosaic virus. The gene is transcribed and translated and protein is formed on the outside of the squash. These genes are then inherited. Wild squash, which is not edible, could breed with the disease-resistant squash and become immune to disease. This causes the weeds to disturb the ecosystem because they can breed with no consequence or predators. The wild squash will take up most of the nutrients and area needed for other plants. Other plants will not be able to grow or will grow less in the area. This will cause the consumers of these plants to not get food and they will have to migrate or die. The secondary, tertiary, and beyond consumers will also have to migrate or die because they cannot eat the primary consumers any longer. The risks of the wild squash disrupting the food chain outweigh the benefits of disease-resistant squash. Disease-resistant squash causes more problems in the environment than allowing squash to succumb to disease.